Hello bachcho, let's see the question. So the question says genetic engineering is, so इन चार ऑप्शन में से हमें वो ऑप्शन आइडेंटिफाई करना है जो जेनेटिक इंजीनियरिंग को बेस्ट एक्सप्लेन कर रहा है. So the key concept in this question is to first understand what is genetic engineering. So first of all, genetic engineering is a technique of gene modification. So we can use genetic engineering to alter the DNA of an organism's genome. It is also known as gene manipulation because with the help of genetic engineering, we can manipulate the gene or alter the gene according to our own interests. Now, if we see the natural phenomenons of gene modification like mutations or crossing over, all these techniques occur randomly. But in case of genetic engineering is a more specific or a more targeted technique of gene alteration. So, with the help of genetic engineering, we can alter the gene of our own interest or introduce the gene of our own interest into a different organism. So, genetic engineering is a more specific or a targeted technique. Also, genetic engineering is an artificial method. So, genetic engineering does not happen naturally. It occurs due to manual interventions. Now, if we look through the basic steps of genetic engineering, then the first step involves the isolation of the gene of our interest. For example, if we want to introduce the human insulin gene into a bacteria for the production of insulin by the bacteria. So in that case, the gene of our interest will be the human insulin gene. Now this gene needs to be inserted into a vector. So the gene of our interest then needs to be inserted into a vector. So in this case, the vector will be a bacterial plasmid. So we can first isolate the plasmid of the bacteria into which we want to introduce the gene of our interest, and then we can insert the human insulin gene into its plasmid. So after the insertion of the gene of interest into our vector, this vector is then introduced into the host organism. So the host organism in this case is bacteria. So now we can see that the human insulin gene, which was responsible for the production of insulin, human insulin, is now introduced into the bacterial genome. Therefore, the bacterial genome can now produce human insulin as well. So in this way, the gene of our interest is introduced into a different organism and we produced a recombinant. So a recombinant is any organism which has DNA of two different organisms. So that's a recombinant organism. So this is how genetic engineering basically takes place. These are the very basic steps of genetic engineering. Now, if we come back to our options, then we see that genetic engineering, the first option says that study of extranuclear gene. So this option won't be correct because it is not just the study of extranuclear gene. It is the study of all types. It's the manipulation of all types of gene. The second option says manipulation of genes by artificial method. So this is the right option because in genetic engineering, we manipulate the gene according to our own needs. So it is gene manipulation and it is an artificial method. So genetic engineering is manipulation of genes by artificial method. The third option says manipulation of RNA. So this won't be correct because RNA is not manipulated in genetic engineering. Also, in the fourth option, it says manipulation of enzymes. Again, in genetic engineering, we do not manipulate enzymes, but we manipulate the gene of an organism. Therefore, our final answer for this question is option 2, that is manipulation of genes by artificial methods. So this is our final answer. Hope you understood it well. Best of luck. Thank you.